Okay, awesome. Great. Okay, so basically what I'd love to tell you about is how do we figure out the circuitry of human disease? How do we understand medicine from a completely different and much deeper mechanistic perspective? And how do we use all of this to truly transform ultimately the human condition, transform therapeutic development, transform the way that discovery is being made? So I don't know if you guys can see, but hopefully you can. Okay, perfect. Okay, so medicine and biology have really come a long way. So, if you look at the Harvard hospitals today, or if you look at sort of ancient Greece, there's a dramatically different scene. So at the time, people used to treat patients from the outside. The doctor, or in this particular carving, perhaps a god, was looking at the patient, everybody was observing, and you basically had the priestesses, and actually coming to think of it, maybe not much has changed compared to the godlike venerance of the doctors at the time. But what I would like to point out is that this was not really getting inside the problem. The technology revolutions of the Renaissance is what allowed medicine to start being dramatically transformed. And then the ability to go in and observe individual organs, to observe individual cells, individual neurons, and to start understanding the cellular pathology that underlies human conditions. So what used to be thought of as simply dementia before Alois Alzheimer gave his name to what is now known as Alzheimer's disease, people just thought that, with old age, you would just lose your mind, and that was normal. But what Alois Alzheimer recognized using these extraordinary powerful microscopes at the time is that the cells from Alzheimer's patients showed neurofibrillary tangles and amyloid beta plaques that have become, since, of course, both the definition and the hallmarks of Alzheimer's disease. And technology has really come a long way since then. So beyond just one or two biomarkers, we now have hundreds of genetic loci that are able to tell us where the causal contributors to Alzheimer's are, starting with ApoE4, BIN1, CR1, CLU, PICALM. There's such a diversity of molecular functions that are starting to be revealed. So that's the first dramatic revolution in addition to the microscope. And then the second one is the ability to not just look at genetic causal components, but to start understanding how those causal components might be acting through the systematic mapping of individual cells from patients and controls at unprecedented resolution. This is a figure from our own lab, something that was just published in Nature just a couple of weeks ago. And what this shows is 430 donors, 2.4 million cells, and every dot here is 20,000 variables, each of which is measuring the expression of a different gene. So we went from having 2 variables on 4 cells on the bottom left of these slides to having 20,000 variables on 2.4 million cells across 430 patients on the right side. And that's what takes us to how can we actually start interpreting these data sets. And by the way, is the sound okay? Do you want me to change anything on my side, or are you guys still good? Okay, good, perfect. Okay, so what I want to get to is that there are really 3 major paradigm shifts. So yes, everyone is talking about AI right now, but there's really 3 major paradigm shifts that have transformed and are transforming medicine. The first one is that this transformation with massive, massive data sets is basically moving us from hypothesis-driven research, where the researchers spend a lot of time formulating their hypotheses, gathering data sets, and ultimately, after a lot of thinking before, do one very small targeted study that gets a yes-no answer as to whether the hypothesis was supported or not supported. And the problem with this is that it's highly biased, and there's generally very little novelty. You have to integrate a lot of knowledge before you even start, but there's very little surprises. You're not going to go off and find completely new things. What we're facing now with this deluge of data sets is a shift to data-driven research, where you basically shoot first and ask questions later. So you gather tons of data, you build systematic data sets, build massive resources. There's massive data sharing that enables the whole community to work together, and really comprehensive views. And from those, very often come insights that we would have just never expected before. The second one is that we went from an epidemiology over thousands of years that is very correlation-based to causation. With correlation, you see that countries that consume more chocolate also have more Nobel Prizes. You see that people who spend $7 on their Starbucks coffee every morning have better health outcomes. Is it that the chocolate leads to the Nobel Prizes? Is it that the coffee leads to better health? Or is it that perhaps if you can afford a $7 coffee every morning, you can also afford better healthcare, better lifestyle, better nutrition more broadly? And the challenge with epidemiology is that these correlations are very hard to untangle. By contrast, with genetic variants that well precede the disease outcomes, these arrows are now unidirectional instead of bidirectional. And we can go from polygenic risk scores and perturbation experiments to understand really causal factors, both through genetics and through these perturbations. So that's dramatically different, because with causality, we know where to intervene, we know what to edit, we know what to rewire. And the third major transformation is that we're going from this classical machine learning and data analysis, where there was a new methodology for every new question, where the human scientists would do all the thinking. We were trying to minimize the number of parameters and build very targeted models, to a complete paradigm shift with generative AI and deep learning. And many people are saying, deep learning is just neural networks repackaged. We had the same thing in the 60s. But that's not true at all. What we had in the 60s is indeed classical neural networks. But what has really changed now is the concept of representation learning. The concept that we can actually have the AI truly understand hierarchical, complex, multimodal concepts that we can build through self-supervised learning, foundation models that are able to capture more and more true deep understanding about the subtleties of human language, of chemistry, of biology, of protein structure, of gene expression patterns, of cellular state, of patient trajectories, of cognitive states, of mental health, and so on and so forth. So we are now using AI as a partner for the discovery, rather than just, hey, tell us what the correlation is between these variables and just find some predictive function that predicts X, Y, or Z. So we truly have these foundational, multimodal, hierarchical, deep insights into the data. And that allows us, the combination of these technologies allows us to now start understanding really the mechanism of what causes disease. So we can start with genetic variants that we know plays a causal role, but we don't know how they act, and then translate that into very specific mechanistic circuits that basically tell us what in the genome, in the epigenome, in a specific cell type, in a specific gene, in a specific control region, in a specific regulator, what is the path through which this genetic variant is ultimately leading to a circuit change, a network change, a cellular change, a molecular change that ultimately manifests at the level of cells and, of course, organisms. So that's the path that we're taking in my lab, and that's the path that many others in the field are taking, where we're starting with causal loci, we're figuring out their mechanisms, inferring through massive data integration, driver genes, regions, and cell types. And now that we have these drivers, we know where to intervene, so we can then go in and validate our predictions in both human cells and in animal models. So to give you a sense of what do these circuits look like, here's a region of genetic association, and you can basically expand that out. And instead of just saying, oh, there's something going on here, but I don't know what cell type, what regulator, what target gene, etc., you expand this out, And you say, okay, well, there's exactly 4 genetic variants in this region. These variants are sitting in enhancers that are active in those tissues and cell types. They disrupt, either directly or indirectly, very specific regulatory motifs, these sequence patterns that are recognized by very specific regulators, which are then forming a network upstream of the circuitry and ultimately targeting downstream target genes that are then the effectors of the function. That allows us to completely open up completely new entry points for disease manipulation, because we know that those genetic variants are causal, and we can use the circuitry to understand what are the points of intervention. Is it this gene? Is it that regulator? Is it this cell type? And so on and so forth. So using this approach, we can now tackle some of the greatest challenges in our society. So you've seen the transformational impact that the GLP-1 inhibitors are having on human health with the reduction of eating. And the strongest genetic association with obesity, however, does not impact the brain. What it impacts is this one locus, this one genetic locus that we dissected in a paper in the New England Journal of Medicine more than nearly a decade ago. And what you can see here is that this region, the strongest genetic association, is in fact perturbing a single letter. We can trace down that entire association down to a single letter from 81 common variants that are spanning 50,000 nucleotides. We can go down to one change from a T in this AT-rich motif to a C. This AT-rich motif is normally bound by the ARID5B regulator, AT-rich interaction domain factor 5B, that normally binds here and represses this enhancer and therefore shuts down this region. But when the motif is broken, this repressor can no longer bind. This enhancer gets de-repressed and turns on 2 genes that are sitting 1.2 million nucleotides away and 600,000 nucleotides away, IRX3 and IRX5. And those genes were previously unrelated to obesity, but what we have shown is that they are master controllers of not whether you want to eat or not, but instead what you do with the calories. And specifically, they shut down the process of thermogenesis or heat generation, whereby the mitochondrial membrane is depolarized, causing a loss in the proton gradient and ultimately loss of energy as heat. So that basically means that every calorie that I do not burn through exercise will then be stored into fat, into white adipocytes, into lipids, these very rich molecules that ultimately turn into obesity. Why are we so excited about this? Because by having the circuitry, of course, we can understand the mechanism, and that's important, but much more importantly, we can start having points of intervention. We can basically say, can I upregulate or downregulate the target genes, IRX3 and IRX5? Can I upregulate or downregulate the upstream regulator, ARID5B? Can I use CRISPR-Cas9 genome editing to rewire the cells of obese individuals from a C back to a T and then restore the natural repression by ARID5B of this enhancer? And the answer in every one of these cases is that it works like a switch. You can go back and forth between lean and obese phenotypes at the molecular level, at the gene regulatory level, at the gene expression level, at the cellular level, and at the organism level. So through a series of experiments in that paper, and it's a very, very long supplement, we basically show that you can take primary adipocytes from risk individuals with a C allele where the motif is broken, restore that single letter from 3.2 billion letters in the human genome, go and make one edit, and then restore thermogenesis, the ability of primary adipocytes to actually burn calories by a factor of 7, both in the control and the isoproterenol stimulated condition. When you do the intervention at the organismal level and you increase or decrease the expression of the IRX3 gene, in this particular case, it's de-repressed in obesity, so we want to shut it off. And what we can see is that by shutting off IRX3, specifically in an adipose promoter construct with a dominant negative IRX3, you can basically see that when mice are grown with this construct, they're leaner to start with. And when you introduce a high-fat diet, normal mice gain weight, but these mice are unable to gain weight. What you can see instead is that they're burning all of their fat stores. Their organs are not white, they're red. They're full of just really healthy tissue and no fat whatsoever. So these mice have basically burned off all of their white adipocytes, and they're instead thermogenizing like crazy. So that's what we want to do systematically. We want to go from a region of genetic association to a mechanism and ultimately interventions, new insights, and therapeutics. So this is the strongest association with obesity. This is the strongest association with Alzheimer's. This variant here increases your risk of obesity by 1 standard deviation. This variant here increases your risk of Alzheimer's by a factor of 10. So homozygous carriers of the E4 allele of the ApoE gene have a 10-fold increased risk of developing Alzheimer's. And what you can see here is that there are, of course, many other variants. So we said, okay, how is it acting? Can we use these techniques? Can we now start studying the molecular consequences of having the E4 variant? And we took both male and female individuals, both AD and non-AD individuals, and both carriers and non-carriers. And what we found is that in the carriers of the E4 allele, regardless of whether they had Alzheimer's symptoms already or not, we found a massive upregulation of cholesterol biosynthesis, specifically in oligodendrocytes. Now, oligodendrocytes are the cells that protect the neurons and protect them by coating the axons with myelin, which utilizes cholesterol. So we see here that cholesterol goes up. And when we look at the myelin, we basically see that the myelin actually goes down. So those cells from the E4 individuals have much less myelin than the controls, but much more cholesterol biosynthesis. So we said maybe they're upregulating cholesterol because they can't get it in the right place. And when we looked precisely within the endoplasmic reticulum, the machinery for export within the oligodendrocytes, we found that healthy oligodendrocytes have these beautiful stripes that are associated with export, but the E3/4 individuals that carry a risk allele, they are basically showing these massive droplets of lipids. So the cholesterol is getting upregulated because we can't form myelin, but it's getting stuck. And we hypothesized that given the apolipoprotein E transporter function of ApoE, maybe it's a fault of transport. And we basically said, what if we restore cholesterol transport using a molecule such as cyclodextrin, which is hydrophobic and can help the transport? And indeed, we found that these restored not just myelination, both in human cells and in mouse cells, but it actually restored cognition in mouse experiments. So once more, we go from a genetic difference to a molecular circuitry, a cellular phenotype, a point of intervention, and a rewiring. As a third example, we basically looked at response to immunotherapy in cancer, and specifically metastatic melanoma. So metastatic melanoma used to be a death sentence. It basically shuts off your immune system and it basically takes over. However, with immunotherapy, about 50% of the patients are able to restore their immune system and fight off the cancer. So we basically said, can we take responders and non-responders and start looking at their circuitry, start looking at their epigenomics, their single cell gene expression, their single cell DNA accessibility, and their histone modifications. And what we found is that responders versus non-responders were different in hundreds of loci that were basically being shut off And we found the upstream regulator for those, and we used a combination treatment in mouse to basically show that the anti-cancer drugs alone were insufficient and only worked in 50% of the mice. But when you use the combination treatment that restored the epigenomic state of responders, then we basically found 100% efficacy for those drugs. So this is what we want to do systematically. This is what we want to enable by understanding that circuitry. So we can basically now start gathering massive data sets to understand this circuitry. And we have basically looked at multiple brain regions with single cell profiling across non-AD, early AD, and severe AD individuals in the progression of Alzheimer's disease. We found dramatic differences between early stage and late stage differences, with extremely cell type specific early changes, but much more shared late changes. We found dramatic sex differences with male individuals showing hundreds, actually thousands of differentially expressed genes from female individuals. 3,000 differentially expressed genes before Alzheimer's, 6,000 differentially expressed genes after Alzheimer's. And we found that male individuals in particular upregulated these myelination pathways and indeed showed much less white matter loss than female individuals that did not. And we can use the time course progression of these millions of cells to start predicting what are the genes that are acting early or late in the disease process. We can also, with hundreds of individuals, 427 individuals in the latest study, we can basically now start asking about more complex phenotypes. Namely, it's not a one-to-one mapping between the individuals that show pathology and those that show cognitive decline. There's a subset of individuals that show plenty of pathology, but no cognitive decline. Thank you very much. So the question is, what distinguishes those individuals? And we were able to find that associated with resilience were specific classes of inhibitory neurons that were very specifically involved here. And then there's a set of pathways that are associated with both the progression of pathology, with neuronal vulnerability, and with cognitive resilience. And those are implicating mitochondrial function, synaptic signaling, epigenomic processes such as cohesin complex expression, also post-transcriptional processes like mRNA metabolism and lipid metabolism, where nearly every single gene involved in this fatty acid beta oxidation is in fact differentially expressed between cases and controls. Focusing specifically on cognitive resilience, we found that yes, neurons played a role, but the largest changes were by far found in astrocytes and oligodendrocytes. And specifically, we used a new module discovery approach to decompose the expression patterns of these hundreds of genes across these millions of cells into a small number of modules. And one of the most striking modules was in fact choline metabolism. So basically, choline was found to be protective against cognitive loss when pathology was present, suggesting that perhaps for those individuals that are starting to show pathological signatures, this might be one way to promote cognitive resilience through dietary interventions. We can also go and study the changes that are happening in the immune cells of the brain, the microglia. And what we're finding is that there's a very specific shift from homeostatic microglia to inflammatory microglia in Alzheimer's. And we can reverse this shift to basically bring the inflammatory microglia back to homeostatic states. And using this rewiring in the brain of mice, we again show that we can restore cognition and we can restore a healthy state. We then focused on the circuitry of the epigenome, and we found that AD individuals show an accumulation of DNA double-stranded breaks and of somatic post-mitotic mutations accumulating in the neurons. And in particular, we found this global mechanism of epigenome erosion that is not found at all in early AD, But in late AD, the cells, instead of being nicely distinct from each other, are basically shifting to this de-identified state. And you can see that the closed regions are opening up and the open regions are closing up, where the epigenome is simply flattening out. So that gives us another entry point to understand the circuitry. We also have a lot of new insights, for example, that ALS, a motor disorder, and FTD, a cognitive and behavioral disorder, are in fact affecting the exact same genes in the exact same cells in orthologous cell types between the motor cortex and the prefrontal cortex, suggesting that in fact the same mechanism might be underlying those two seemingly different disorders, consistent with the common C9orf72 basis of these disorders along this continuum of TDP pathology and tau pathology. We're also able to characterize the vasculature in much more detail. We're able to understand how that vasculature is changing in Alzheimer's disease. We're finding with spatial transcriptomics, we can pinpoint, perfect, 5 minutes. Yep. We can pinpoint very specific landmarks where these changes are happening. We can look at schizophrenia and start understanding the specific circuits underlying the genetic factors involved in schizophrenia and the upstream regulators. And we can apply these lessons to psychosis and find that, in fact, it is associated with major repression of neuronal activity rather than overactivation during those psychotic episodes. And that it has something to do with long-range communication, specifically in the layer 2-3 long-range acting neurons. We can also understand the mechanisms of addiction and the specific D1 and D2 receptors underlying all this. So study after study, trade after trade, obesity, Alzheimer's, we can now start understanding what are the hallmarks of disease, what are the recurrent changes that are happening between different patients, and start mapping those changes to specific cellular neighborhoods and specific mechanisms. And we can use that to now start developing next generation therapeutics. We can actually start building, using the next generation of AI tools, joint latent representations of protein function, of cellular state, of biomedical text, and start reasoning with human interventions to those latent models. So in my introduction, I talked to you about this dramatic shift in generative AI and in representation learning. We can now take these knowledge graphs that connect all of these different molecular variables together between genes and diseases and phenotypes and drugs, and start asking through graph neural network latent embeddings, how are these in fact distinguishing different classes of proteins? How are these classes of proteins related to the structure of those proteins? How is that related to the cellular functions and the latent embeddings of these cells? How is that related to the latent embeddings of patients? And how those embeddings are relating with the gene expression changes of these patients? We can now start going into human emotional wellness and start asking, based on the responses of these patients, can we build latent representations of emotional wellness and emotional struggles that humans undergo through these surveys? We can now use AI and large language models to now start building these interactive latent embeddings of protein function. Here I'm showing you a map of 20,000 human proteins, where all we did was take the biological description of these proteins and we fed them into a large language model to study their embeddings. And what we can see is that this is able to uncover the diversity and the continuity of cellular function. And we have now built a tool that we call Mantis, which is an AI-powered interactive visual data science workbench. We don't want to just look at these, but we want to start interacting. We want to be able to search. We want to be able to interact with an AI agent, an orchestrator, and a narrator. And we're able to now start understanding the hierarchy of these and start interacting with these computer Jupyter sandboxes. So the future that I see is one where we can basically build these reasoning, interactive, user-guided, AI-powered agents for drug design and for therapeutic design that understand chemistry and proteins and biology and 3D structure and human language, that we're able to chat with and interact with. And this is basically what we're building, this extraordinary collaboration with Brad Penteluta and Marika Zitnik, where we are basically now guiding a large-scale interventional model, guided through this complete pipeline from genomics all the way to therapeutics to understand the pathways, understand the 3D structure, understand where to intervene, and optimize the chemicals. So that's the path ahead, where we need to transform the way that pharma is thinking. We need to start personalized medicine. How? By uncovering the common pathways and mechanisms underlying these disorders and using AI, biology, chemistry, and synthetic biology to start reversing them systematically. So that's the path forward, where we can use AI as a partner in discovery, in truly gaining new insights and understanding, and in rewiring those and moving forward in a Google Maps for navigating this knowledge. I'm truly privileged to be here and to be speaking to you and to be reporting the work of so many amazing collaborators and folks in the lab across both the computational and the experimental sides, and truly generous support by both NIH and a large number of institutions. So that's where I'll stop. And thank you so, so much.